This is From Our Neurons to Yours, the podcast from the Wu Tsai Neurosciences Institute at Stanford University, bringing you to the frontiers of brain science. Today on the show, inflammation and Alzheimer's disease. For decades, the big hope for treating Alzheimer's disease has centered on finding a way to clear the brain of the sticky amyloid plaques that define the disorder. And in just the past few years, multiple new drugs have come out which are, by all accounts, quite effective at doing just that. But unfortunately, what should have been a celebration has become a bit of a disappointment. A few episodes ago, we had Stanford neurologist Mike Gracious on the show, and he pointed out that while the new drugs do reduce plaque buildup, the clinical benefits reported are minimal, essentially undetectable to patients and their physicians. This has led to a fierce debate among the experts. Some argue for abandoning the focus on amyloid altogether. Others maintain that genetics prove amyloid is an important player in the disease. Maybe we're just intervening too late. In any case, the Alzheimer's field that had long been unified around the amyloid hypothesis is now divided and desperate for alternative approaches. Today's guest is Kati Andreasson, a physician-scientist at Stanford whose lab has spent the past 2 decades patiently following scientific clues to understand the links between aging, Alzheimer's, and inflammation in the brain. Initially, Andreasson struggled to get funding for her research, as most people were fixated on amyloid. But she and her team have meticulously pursued a series of biological questions that have succeeded in painting a new vision of how Alzheimer's arises, as an age-related energy crisis affecting the brain's immune cells, the microglia, and other key support cells called astrocytes. They've shown that reversing this metabolic crisis in mice bred to develop Alzheimer's disease can restore both memory and cognition. Most recently, they've made the kind of stunning discovery that targeting age-related inflammation in the peripheral immune system, so not the brain at all, can also treat Alzheimer's symptoms in mice. That's pretty far out. Now, we should also be clear, Alzheimer's has been cured in mice many times, so we need to wait for human trials before getting our hopes too high. But at a time when Alzheimer's research is facing a long sojourn in the wilderness, I was particularly excited to talk with Kati because her work is such a good example of the importance of curiosity-driven science, even when it goes against the grain. And plug for the Knight Initiative for Brain Resilience here at Wu Tsai Neuro, which funds some of this work. That's what this initiative is all about, cultivating new ideas about how we can keep our brains healthy and prevent neurodegenerative disease. So I asked Kati to join us on the show to share her scientific journey and help us understand this promising new battle line in the fight against Alzheimer's disease. Kati Andreasson, welcome to From Our Neurons to Yours. It's so great to have you on the show.
Thank you. It's a pleasure.
We've had a few conversations on the topic of how the thinking in the field of Alzheimer's research has been changing over the past few years. There has been this incredible focus for many years in Alzheimer's research on amyloid plaques, these gunky accumulations of protein that we see in the brains of people with Alzheimer's. And a lot of resources and a lot of time have gone into figuring out how to clear those out of the brain. We now have drugs that seem to do that pretty well. But in the words of your colleague Mike Gracious, who was on the show a few months ago, those drugs in the end for patients seem to be kind of underwhelming. Do you agree with that assessment?
Yes, I think from a clinician standpoint, they are underwhelming. They are very expensive. So yes, I agree. We really need something much better and much more efficacious.
Well, you got an award earlier this year from the Alzheimer's Association for most impactful paper in the field of Alzheimer's research for the past 2 years. So congratulations on that.
Thank you.
And it reflects this different approach that you and your research group have been taking over the past couple of decades to thinking about the role that inflammation and more recently metabolism in the brain might be playing in where Alzheimer's comes from. I mean, I want to talk about this research journey. This started 20 years ago. But before we dive into that, I mean, can you tell us a little bit broadly about what is inflammation and why does it happen in the brain? Why is it so bad for the brain?
Yeah, I mean, inflammation is complex. So we absolutely need our immune cells, which give us inflammation, to defend us from viruses and bacteria. And we also need our immune cells to take care of all the cellular waste that we make in our bodies every day. So if you think about it, we make, hundreds of billions of red blood cells, and they have a finite life. And immune cells, specifically macrophages, gobble these up on a daily basis. It's a huge amount of work. And so that's just one example of an internal function of the immune system, is just to keep everything humming along. We have immune cells in all our organs. My lab is really focused mainly on the macrophage population. This is the first responder. It's like, the firefighter who goes out there to put out the fire. If there's an infection, the macrophage is there, tries to clear it. And macrophages operate everywhere, in your liver, in your gut, and in your brain. And they have a special name in the brain. They're called microglia. And they are, tremendously important. And in the last couple of years now, we've really begun to appreciate the importance of the macrophage in our health, and how as they decline, so do we.
Right. With aging, those macrophages kind of get exhausted.
Yeah, yeah.
I just want to go back for a second. So this is something I'd love for you to help me clear up. You said that the macrophages are one of our first responder immune cells throughout the body. And in the brain, they have a special name, the microglia. Why do they have a special name in the brain? Are they the same cells, or is it a different cell with the same job?
It has the same job. So it basically keeps the brain clear and pristine, gets rid of stuff like amyloid. It also gets rid of dead synapses. They're called microglia, I think, because they're smaller than, a macrophage that you find, in your liver. They also look a little different. But this is the beautiful thing about macrophages, is that they adapt just like we would. Like if we're in a different environment, if we're sitting in the liver, they have a certain function and they have a certain size and shape. If you're in the brain, which is, completely different kind of organ, where you have, axons, you've got neurons, you've got neuron processes, they sort of adjust, and they have a very different shape, and they also function in a different way. They're just continually sensing, they're putting feelers out, just making sure that the local microenvironment is kept pristine, so that, of course, our neurons can work.
Right. I mean, they do so many things. It's sort of like a mix between maintenance and custodial work, and, as you said, police and firefighters, right? If there's an infection, they're going to be involved. So just absolutely critical to keeping our brains clear and healthy and clean. So I think that many people have probably heard about the problems with brain inflammation. This is something that comes up, for example, talking about long COVID, that there seems to be elevated inflammation in the brain, comes up with aging. I mean, the inflammation is clearly a big problem. What is inflammation, actually? What does that mean?
So, yeah, I mean, when we think about inflammation, we think about the detrimental component of inflammation. And so this is something where you get a lot more production of inflammatory factors that have bad effects on the target cells. For example, if you think about aging, one of the most common age inflammatory diseases is frailty, muscle wasting, loss of strength. And that is the effect of inflammation on the muscle. So you lose muscle mass. Another example is a very common syndrome, atherosclerosis. Like so many people have heart disease and vascular disease. That's also inflammation, because you have within the walls of the blood vessels, the arteries in particular, you get deposition of cholesterol, and then in come the macrophages. They're trying to clear it. They're trying to do their job. But there's so much of it, of this cholesterol, that the macrophages actually get sick. And they become inflamed. And so then they recruit other cells. And so that's bad inflammation. There's so many examples. Metabolic syndrome, where we eat too much and we eat the wrong things.
And that's sort of obesity and diabetes and so
Yeah,
on.
exactly. And the tissue, it's called the adipose tissue. So this is the tissue in the belly that gets bigger and bigger. It's full of macrophages, and these macrophages are just churning out all these nasty cytokines and chemokines. So these are immune factors that signal to the rest of the body. And one of the side effects, unfortunately, is development of insulin resistance. And that sort of predisposes people to diabetes. So there are many examples of age-associated inflammatory diseases. Chronic arthritis, you have chronic inflammation in your joints. Periodontitis, big one of the gums, a huge risk factor for Alzheimer's.
Oh, interesting.
So, yeah, there's just so many inflammatory diseases that get worse with aging unless we can sort of fix it, which diet and exercise really are the current best therapies. And they can work actually quite well, but we don't have that magic pill yet.
Right. Well, one of the things that's so interesting here is we talked a little bit a moment ago about the idea that, well, Alzheimer's disease involves these buildup of these amyloid plaques in the brain. So if we could clear those out, maybe we clear up the disease. So far, it doesn't seem like it works that way. And one of the observations that many people have made and that you've been following is, well, why does Alzheimer's happen as we get older? And maybe the amyloid, maybe it plays a triggering role. Maybe it's something that is actually more of a side effect, that amyloid is also caused by these processes that go on during aging. But we somehow need to get to the root causes so that we can figure out when do we need to intervene and what is the best target. So in pursuing inflammation, you've told me that the inspiration for your work came in the early 2000s when researchers in the US and Canada discovered that people who regularly took these non-steroidal anti-inflammatory drugs, which is the fancy name for drugs like ibuprofen or Advil, for several years had a significantly reduced risk of Alzheimer's disease. Can you take us back in time? I remember that being a big deal, but I want to know what was your reaction to seeing that research come out?
Yeah, that's kind of what got us started. So these studies were done across the globe. Actually, there's a study, one of the studies was done in the Netherlands where they have nationalized medicine so they could track everybody. Another one, which was a very compelling one, was done out of the VA. But basically, what they looked at, and this is super important, these were cognitively normal people. So they had absolutely not a whiff of anything. And they would take NSAIDs for aches and pains. It didn't even have to be every day. It's just the use of taking these NSAIDs.
And the NSAID, that's the sort of abbreviation for
these
non-steroidal anti.
Yes.
We don't want to keep saying non-steroidal anti-inflammatory drugs.
Yeah, it's too long.
NSAIDs.
Yeah, but things like, ibuprofen, indomethacin, not Tylenol, and not aspirin. So those are not quite the same. But people who would take those, it would take a while, and this was best illustrated in the study out of the VA in 2008. It took about 4 to 5 years to actually see this effect, where people had this sudden drop in their risk of getting Alzheimer's disease. It was as big as 25%. Which is, considerable. So that got me thinking, well, maybe inflammation is something here, and of course at the time, nobody wanted to hear anything about except amyloid. And I have all these, sad stories of trying to get grants from the NIH and getting continually rejected and nearly quitting and going into the clinic. But eventually, things did work out, and we started to look at this pathway, and NSAIDs, they block a very particular set of enzymes. They're called the cyclooxygenases, and there's 2 forms, and one of them, we call it affectionately COX-2. Cyclooxygenase 2 is it's like the mother of all inflammatory enzymes because it makes something called a prostaglandin, and there are several prostaglandins. They do very different things in the body, but one of the prostaglandins is the immune inflammatory modulator, and that's called prostaglandin E2. And so that's what we've spent the last 20 some years looking at this pathway and how that would be involved in driving Alzheimer risk.
So before we dive too deep into that, you've done this amazing work breaking down this pathway, but I want to just dwell on the NSAIDs for just a moment more. You mentioned that the NSAIDs are distinct from aspirin and from Tylenol. What exactly are NSAIDs? What are they doing?
So NSAIDs are blocking both cyclooxygenases. So there's COX-1 and COX-2. Tylenol, it's still not clear how it works, or at least I haven't been keeping up with it, but it's not really a COX-1, COX-2 blocker. And aspirin is just very specifically a COX-1 blocker. And the specificity of what these drugs do really has to do with in what cells you are making the COX-1 or COX-2. And so for a long time, aspirin, and it is very well known that it causes platelets to thrombose. And so that's the benefit, the cardioprotective effects of aspirin. Although this is all now slightly put into question more recently with some new studies.
It's funny. These are such like tried and true drugs, but we're still learning new things about them. Are they all acting on inflammation, or is that something that is particularly true of ibuprofen?
Well, ibuprofen has the benefit that it hits both COX-1 and COX-2. So COX-2 really is the inflammatory enzyme, but COX-1 is also helping out. So it's nice to hit both of them. And so that's why these NSAIDs are very good, for aches and pains. And of course, this preventive effect. I have to say that these early studies, people got very excited about them, and then they started trying NSAIDs in people with cognitive decline, and they did not work.
Interesting.
And that's why the whole field is sort of confused, because sometimes people don't distinguish between the preventive in normal cognition and the potentially therapeutic, and it did not work at all. And once cognitive decline has set in, the ship had sailed.
Right. It's this thing that it's so hard. I think that it's hard for us to understand, and maybe that's just hasn't percolated from the lab into the broader culture. By the time cognitive problems are happening in Alzheimer's disease and many other disorders like this, stuff has been going wrong in the brain for many years already.
So long.
And the brain has so many ways to adapt, right? The brain is really good at making do with what it's got. So these things might be killing off brain cells and synapses for decades, and the brain is adapting and finding new ways of getting stuff done. By the time other people start to notice, or you start to notice yourself, that may be too late. And so one of the things that's so challenging is, how do we catch it early? What are we trying to catch, for one thing? And how do we get there early enough that if we can fix it, it means that we're preventing something that hasn't happened yet? I guess medicine isn't always very good at that. So why was it not the recommendation to just everyone should be taking NSAIDs?
Well, that's a very critical question. It is not a good idea, and people should not go and start taking NSAIDs because they have side effects, and they block, sort of the top of the pathway, which is the generation of the precursor of 5 prostaglandins. There's not just one, but there are 5.
And these are these sort of inflammatory signaling molecules, the prostaglandins.
Yeah, right. So 5 prostaglandins, only one is really involved in inflammation. The other ones are super important. For example, we have something called prostacyclin, which would be blocked by an NSAID. And prostacyclin is super important in allowing your blood vessels to dilate. If you don't have that, your blood vessels are constricting and you get high blood pressure. We have another prostaglandin, it's called PGD2. You don't want to block that either, because that's very important in sleep-wake cycle. So these other prostaglandins, there's the other 4, have very important functions. So by going at the very top of the pathway with the NSAIDs, you're actually blocking not just PGD2, which turns out to be probably the bad guy, but you're also blocking the other 4, which are very important. So it's not a good idea to take NSAIDs. What you want to do is you want to just move down the pathway, inch by inch until you hit the sweet spot. And which is what I think we have now finally done after all this time. sort of figured out, we know it's now PGE2 that's really the important prostaglandin. And then after that, we had to figure out, what receptor. It's like a key in a lock. That's a receptor binds the prostaglandin. But there are 4 receptors for PGE2. So we had to spend many years parsing that out until we finally got the answer. So, yeah, it's a long haul.
Yeah, I mean, from these original observations were in, was it 2001, 2003, I think?
Yeah, something like that. Yeah, that's when I started my lab. Yeah.
So you've been, as you said, you've been sort of working out this pathway. Okay, we know NSAIDs are reducing Alzheimer's risk, but we don't want to be giving people NSAIDs. So what are they doing? You mentioned that they are targeting this particular COX receptor. I think you called it the mother of all inflammatory molecules, which is great. And then, okay, what does that do? So the COX receptor is making these prostaglandins get released that are these signaling molecules. Which one of those is the bad guy? You identified PGE2. It looks like that one is elevated. There was evidence that one was actually elevated in people with Alzheimer's. Is that right?
Yes. And this was actually Tom Montine, who when he was at University of Washington, and he's at Stanford now, though, his lab looked at cerebral spinal fluid from healthy patients and patients with Alzheimer's. And lo and behold, the PGE2 levels in that cerebral spinal fluid were sky high. We were very happy to see that as well. So then it was just a matter of now really drilling down on that pathway and figuring out which of the PGE2 receptors is the bad guy. And we figured that out. And then how does it work?
Right.
What does it regulate? Because obviously receptors, they signal to the cell and the cell makes new genes. It transcribes new genes and makes new proteins in response to the PGE2 signaling. So that's kind of, we've been, the tortoise and the hare. I'm definitely the tortoise, but...
Well, just in time. Just now, amyloid. We not to trash amyloid too much. I mean, it is the thing that defines Alzheimer's disease. It was important to try, but we need new approaches. We need to be thinking about what are the early causes. And man, Kati, biology is complicated. All of these. There's so it is fun. It is fun. All of these different receptors, all these signaling molecules, they all do different things in the cell. I mean, I think for someone who's not in the field, particularly, just saying, how do you break down this puzzle and figure out, okay, which of these things do we target? And I think what you just said is so important. What is it doing to the cell, right? Why are these cells getting unhealthy? And to do that, you need to go through all these signaling pathways, understand what's going wrong, and so on. But I think one of the things that I found so fascinating in grappling with some of the science, which I have to say took me quite a while to go through all this stuff, and I probably still don't understand it, is this question that's not just about what are these molecules with funny names. It's what is actually happening to these cells that play such an important role in our neurological health and in our health overall as we age. So let's get to that a little bit. So we've mentioned a bunch of these molecules, and listeners, you probably do not need to remember the names of all of these molecules. But the point is, there's a signaling pathway that's involved in inflammation, and your team was able to track down what are the signals, what are the receptors, and you could delete that particular molecular receptor in these cells. And in mouse models of Alzheimer's disease, they got better. They improved their memory and cognition. These are older mice with a lot of amyloid, and it really improved their cognition, right?
Yeah, actually, that we haven't actually published that yet. But
I'm sorry.
Sure.
Excuse me.
No, it's fine. It's fine. Yeah. But we did look at Alzheimer mice, but we were not initially looking at cognition because I think at the time we were still pretty green. But we were looking at amyloid, actually. And it looks like if you delete the EP2 receptor, which is the bad receptor that binds PGE2, these microglia in the brain suddenly become super healthy and they can gobble up the amyloid and they can clear it. And in the process, the brain and the neurons become much, much healthier. So that was the first part. And then more recently, we figured out that in aging, if you do the same thing, if you delete EP2 just in macrophages, you get these tremendous effects on cognition. You can prevent age-associated cognitive decline. And I want to do a shout out for a brilliant MD PhD student who basically did a lot of these studies and also was instrumental in getting us down on this track of real mechanism, like how does it really work, and it works actually through energy metabolism of all things. And, he figured this out, and it's just been such a ride to try to figure this out. It's like being a detective, and trying to find the truth.
Right. And to track down, okay, what are we going to do about it? So I'd love to hear, what's your model of why these cells are getting sick, particularly with age? Because I think that the connection here, this doesn't do a lot if there isn't amyloid, and it doesn't do a lot in young mice. But somehow with the combination of aging and amyloid, I think, correct me if I'm wrong here, something is going wrong with this EP2 pathway that is causing these macrophages and microglia to get sick. So at a high level, what do you think is happening to these cells?
Well, you mentioned amyloid and you mentioned aging, and the one thing they have in common is detrimental inflammation. And that's caused in part by these cells, these macrophages and microglia getting sick. Why do they get sick? Well, we can say that with aging, we suffer a decline in our energy metabolism. It's like an old battery that barely works anymore, And so if you're an immune cell, if you're a macrophage, and you're supposed to be, cleaning things up and defending against viruses and bacteria and things like that, you don't have enough energy, you're not going to be able to do your job very well. You're going to have this anemic response. And so what we found that as macrophages age and as microglia age, they lose, key energy processes, and that translates into a steady deterioration in their immune functions. So whereas they should be keeping us healthy, they actually can't clear bacteria, viruses, debris, so amyloid builds up. So it's just this sort of feed-forward cycle that just gets worse and worse. And so that's what happens in aging. And to a certain extent, if you think about amyloid, when it starts to accumulate, is a really irritating substance. It's activates.
Basically a cellular byproduct, right? Like there's an amyloid precursor protein, it gets cleaved, and if it's not cleaned up properly, it starts sticking together and making these plaques.
Yeah, and the cells around the amyloid just really don't like it.
Right, it's like plaque on your teeth almost.
Yeah, it's like periodontitis. Constant infection. I keep coming back to periodontitis, but it's such a simple example of chronic inflammation that is linked to all sorts of bad stuff.
And our microglia are no longer brushing our brains appropriately.
Yes, exactly. Right. So kind of how we think about it. And the other thing I wanted to highlight, the human genetics that has been done over the last 10, 15 years, where you can look at people's genetics, look at their genomes, has clearly come up with this fact that variants of immune genes are actually the most associated with Alzheimer's. So that was a huge advance, I think, in the field. And this is relatively recently. So that's another reason why we have to think about inflammation, not just for Alzheimer's, but other similar inflammation-related diseases.
There are a couple of things in this. I don't want to get too sidetracked because I think that we've got the major idea here that with age, our immune system is getting tired, sort of exhausted, is not able to do its normal cleanup duties, which allows more amyloid to build up. The amyloid causes more inflammation in these sort of cranky, tired out immune cells. That makes the immune cells more inflammatory and less able to clear up the amyloid. And so it just gets worse and worse. But there are a couple of little things in there that I thought were kind of cool, which is, tell me if I understood this correctly. You're thinking that this prostaglandin signaling is sort of normally there to switch cells into a sort of energy conserving, sort of low power mode that actually helps in an acute crisis like stroke where there's oxygen deprivation. Did I read that right?
Yeah.
So it's sort of like there's this low power mode that cells can go into that can help them survive like in an emergency. But that with aging and this sort of general exhaustion of the system, they get sort of stuck in this low power mode all the time, which starves the cells. And that's one of the things that ages these immune cells and makes them so cranky and inflammatory.
Yeah, that's a really good way to put low power mode. I'm going to use that. That's a very good way to explain this. Thank you.
Okay, so these cells get stuck in their low power mode. They're no longer cleaning up. They're getting more inflammatory. Amyloid is maybe a trigger for that. And then it just sort of snowballs into this vicious cycle. So I think you mentioned that it was hard to get people's attention to some of this stuff early on because people were so focused on amyloid. And you even said it, there are a lot of grant applications you sent in on this that did not get funded.
Yeah,
must have been
so
really
discouraging.
difficult.
Yes.
And you said you were thinking of just going back to medicine and getting out of research entirely.
Yeah, I was starting to, look at the local hospital and they needed a neurologist. But thankfully, I think there was a very, very kind soul at the NIH who took pity on me, and because this was my very first, grant, and somehow gave me $50,000, which kind of allowed me to limp along, and then things changed for the better. There was a program called the Beeson Award, and that really actually made all the difference, given by the American Federation for Aging Research. So I think if it hadn't been from that, I would probably be a doctor somewhere.
Well, it really does seem like things have changed. And I want to get to the really interesting newer work on where the microglia work is headed as far as developing therapeutics.
Actually, this is the other interesting discovery we've been making, is that, for example, with the EP2 pathway, we don't actually have to target the microglia. We can inhibit the EP2 signaling in just macrophages in the periphery.
And that's so surprising because there's been this model forever that there's this blood-brain barrier and the immune system from the periphery is too rowdy and rambunctious to be allowed into the brain. So they're separate. But if you can target this system in the periphery and have effects on the brain, what does that mean about our whole model of how the brain's immune system works?
Yeah, that's the thing. We're a bit of the odd man out here, but I really believe that we could probably target the periphery and not even have to get in the brain. Just based on a lot of data that's now coming out, there's this concept that the periphery does influence brain function. The question is, could you exclusively target the peripheral immune system to get benefits and brain resilience and things like that? And I think it's possible, at least in experimental models. Whether that will translate into humans, we just don't know.
Yeah.
But
and you've got funding now from the Knight Initiative here at Lucid Neuro to do some of that
work. Thank you. Yes. We really think that the peripheral immune system, especially macrophages, are huge drivers of brain health.
That's really exciting. Is this getting translated into clinical trials yet, or is this still pretty early stage trying to target these prostaglandin inflammatory pathways?
Yeah, that's tough. That is tough. I think the thing that we've been working on that's actually much closer to translation is in the astrocytes, which is, again, an immune pathway, and it's actually an immune pathway that can be regulated by PGE2, so that kind of puts everything together. And that is the enzyme in the astrocytes called IDO1 that makes a metabolite called kynurenine. And that is actually, if you think about translation, probably much, much closer because fortunately, there have been a lot of IDO1 inhibitors developed in the cancer field. And so that, to me, is probably much closer than anything targeting the other to EP2 at this point.
We've been talking mostly about microglia, the brain's immune cells, and maybe sort of their equivalents outside the brain, the macrophages. But in 2024, you had this amazing study, and this is what you won this award for earlier this year, looking at a parallel process going on in Alzheimer's disease that affects astrocytes, which are another type of brain cell that's really important for supporting neurons. I was trying to think of a good metaphor for this, but one of the things they do is they sort of keep neurons fueled up, kind of like a pit crew for a fancy race car. Neurons take a lot of energy, and so the astrocytes, one of their jobs is to provide extra energy for the neurons. So can you tell us first what got you thinking about astrocytes and their metabolism? And then I'll ask you about the results.
Well, So this pathway that where you take the tryptophan, which is an essential amino acid, you have to eat it, and it gets converted to this molecule called kynurenine. And this is actually a very important immune pathway. So it's in macrophages, it's in T cells, B cells. And we had actually been examining that pathway in macrophages. There's a long backstory here where we had, anticipated one outcome and we found the exact opposite, which pointed us to a completely new cell, which was not the microglia or the macrophage, but the astrocyte, which happens to also have a lot of this particular enzyme. And there's not a whole lot known about what this does in the brain. There had been some links with schizophrenia because of a downstream metabolite of kynurenine. But so we just shifted. And this is the important thing with science. You have to be flexible. You have to be open. So we started looking at astrocytes. And again, this is Paras Minhas, our brilliant MD PhD student who spearheaded this work. Turned out that in astrocytes, there was actually also a metabolic component. Just like we had been studying in macrophages and microglia, we had really zoomed in on the metabolism and how that influenced function. So in the astrocyte, it was actually very similar, except in this case, by changing the astrocyte metabolism, since astrocytes have a very key role in supporting neurons metabolically. So astrocytes will give neurons something called lactate, which is an energy molecule. So by changing the astrocyte metabolism so that they could give neurons more lactate, we discovered this very new function of this immune pathway in the brain. And basically the punchline is that we looked mainly at amyloid and tau models. So these would be models of Alzheimer's disease pathology.
In mice.
In mice, yeah, in mice. In those models, what happens is the astrocytes just get exhausted, similar to what we saw in aging macrophages. They just get tired and they don't make enough lactate, basically, to give to the neuron. But if we inhibit this particular pathway, this IDO1 enzyme, and we reduce its product called kynurenine, by a very complex signaling mechanism, which is all described in detail in the paper, we can actually get them to make more lactate and thus rescue neuronal metabolism. And I think the exciting thing is that this could be a third rail, so to speak. Alzheimer's is characterized by amyloid. First you need amyloid, and then you get the tau, and the tau spread through neuronal circuits. But at the same time, you're seeing a decline in energy metabolism in the brain. So glucose is not being metabolized properly, and the brain basically is in an energy depleted state. And so what we are doing here is we're basically taking that different manifestation of Alzheimer's and fixing it with, at least we did that in the mice.
And you were able to, it improved their memory, it improved their cognitive function.
Yes, it moved everything. Yeah. And the interesting thing is at the same time that we were rescuing these neurons, they're working better, cognition is back. At the same time, we also found that we reduced amyloid. Why? We don't know. And that's what we're working on right now. And also reduced this pathologic form of tau. Why? We don't know. So that's the next set. That's what we're working on. Well, actually, first we're trying to get some NIH funding for this.
Right. If any NIH program directors out there are listening to the show.
Yes, hopefully.
Well, yeah. So I know we've got to close, and I just want to close on this last thought that it feels like we've taken these really important steps back from focusing on the end product of what we see in late stage Alzheimer's brains full of amyloid plaques and these tau tangles, and taken a step back to look at aging, to look at inflammation, and to look at energy metabolism, these very fundamental things that are setting the groundwork for why does this disease happen with aging? Why does it get so bad? Why are all of these other things going on? Maybe we could just close with, what is your hope for where we are going in the next 5 to 10 years of what is actually going to be a way of helping patients with this disorder to take this back to the therapies?
Yeah, well, I think everybody is hoping for better and better biomarkers. What we need are biomarkers that can tell us 10 years ahead of when we're going to start developing cognitive decline, whether we have something, some problem, like do we have too much amyloid? We do have ways to look at this, but obviously it would be too expensive. So we need more and more sensitive biomarkers that are predictive of future disease. And that is a very active area. There's been huge progress. It's really very exciting. In terms of treatments, I think we need to consider alternative approaches. And that's already being done. People are thinking about targeting inflammation. Unfortunately, the one that people had some hope with, which was targeting this very good inflammation receptor called TREM2, the clinical trials didn't show efficacy. That's very unfortunate. And I think there's a lot we can learn from those trials, and we have to keep plugging away at that. But then the final thing, which I would argue, is that maybe we should think about targeting brain metabolism by repurposing these cancer drugs, these IDO1 inhibitors that have already been extensively tried in cancer. So I am hopeful, as long as we can keep science funded in the United States, and it's not a political issue.
Right. Hopefully Alzheimer's is not a political issue. Hopefully we can all agree that it would be nice to treat Alzheimer's.
I would hope, because, politicians get Alzheimer's. So, maybe they will see the light.
Well, thank you so much, Kati, for coming on From Our Neurons to Yours. This has been fantastic. And I hope we can have you back as more of these exciting discoveries come out.
Yeah, thank you, Nick. Thanks. It's been a pleasure. Thank you so much.
Thanks again so much to our guest, Kati Andreasson. She's the Edward F. and Irene Thiel Pimley Professor of Neurology and Neurological Sciences at Stanford Medicine. To read more about her work, check out the links in the show notes. If you're enjoying the show, please subscribe and share with your friends. It helps us grow as a show and bring more listeners to the frontiers of brain science. We'd also love to hear from you. Send us an email at neuronspodcast@stanford.edu and let us know what's working for you about the show, your favorite episodes, or questions you'd like to have us answer. Quick programming note, we're going to take a few weeks off, but we will be back in 2026 with new episodes from the frontiers of brain science. From Our Neurons to Yours is produced by Michael Osborne at 14th Street Studios, with sound design by Mark Bell. I'm Nicholas Weiler. Until next time.